little bit and not give you a update on angiogenesis in the typical sense, but rather talk a little bit about the question of vascular networks and how they can be made by combining processes of angiogenesis with some other processes, and I will give you some example why that is really necessary. First of all, you all know that the microcirculation and that these are the vessels which are most dynamically changed by angiogenesis are obviously the location of vascular control, because the flow resistance is very high in these very small vessels. And on the other hand, you have also to accept that you have small vessels, but then you have a system of increasing hierarchy, and only with small vessels you will never make a proper vascular network. Even if the smallest vessels are the vessels of exchange and regulation, the larger vessels need to be there to distribute the blood flow. And when you consider the different tasks in such a vascular or microvascular network, then you could say, okay, we need low diffusion distances to every tissue cell, and due to the very limited oxygen diffusion capacity, these distances are in the range of 20 to 100 micrometers, so that's a kind of fundamental restriction. And to have these low diffusion distances, you need angiogenesis driven by hypoxia, for instance. On the other hand, you need also a homogeneous perfusion, which requires diameter adaptation. I will come back to that. And you will need, for instance, for the heart, here is a slide from a coronary talk. You need an adequate coronary reserve, but the same in other vessels, and therefore you need some kind of vascular reserve. So the vessels have to be able to change their diameter by tone. So in principle, when you consider that, you have three different modes of reaction, and these different modes also correlate to different time scales. The tone regulation is usually effected over very short periods in time, like seconds to minutes, whereas the angiogenesis is obviously a much slower process, and the same is true for the diameter adaptation on a structural basis. And I will now try to integrate that a little bit into the question how these different mechanisms, especially angiogenesis, pruning, and vascular diameter adaptation, can be combined to create functionally adequate networks. So let's first have a look into what people normally think vascular networks look like. And if you go to Google, you get typically graphs like this, also in text. Many textbooks, they look like this. This is a very engineer-driven approach. Every vessel has the same length. Every pathway to a capillary has the same structure. So it's a 100% symmetrical network. It is an engineer solution. It might be a heat radiator on a truck or something, but that's what is very strongly in people's minds. Then you have individual capillaries. This is a Croak model of an individual capillary, which has been very, very helpful and famous in microcirculatory research. And then you have also some, a little bit less, pictures of these kinds of more irregular meshes, but a mesh without any hierarchy, so all vessels look the same. And you can consider what are the pros and cons of these different approaches. If you start with the capillary mesh, the good thing is it has a very homogeneous diffusion distance, so every tissue cell is very close to a nearby vessel. And as you see here, this is from the chorion allantois membrane, but it could also be very similar in the lung. And the chorion allantois membrane of the developing chick is a kind of a lung because it takes the oxygen for the embryo. There you have these kind of capillary meshes. So the pro of a capillary mesh is a homogeneous diffusion distance, but on the other hand, you have a very high flow resistance from here to here because you have only small vessels. And you know the Poiseuille law, which gives the force power dependence of flow resistance to vessel diameter. So if you go from here to here, it's very tedious. Just consider traffic in South London. If you don't have a larger highway, it takes you forever to go to Heathrow. You are close to each individual home, but you don't want that. You want to be fast. So this is not very efficient. And then if you feed the system with high oxygen, then you have an oxygen gradient, and you might have oxygen enough to here, but not anything left somewhere later on the way. So you have an oxygen gradient. The other alternative, or extreme if you wish, are very symmetrical trees where you have a lot of hierarchy. So here you have capillaries, precapillaries, small arterioles, and then you go pretty fast by slightly bigger vessels, but with a lot less flow resistance to this point, and there you have the capillaries. So you have a low flow resistance, but actually the proper distribution of oxygen happens only in the capillary region, so you have a restriction of oxygen supply to the capillary region. You have systems which are very hierarchical, like here again in the CAM, but they are different because the capillaries here are everywhere, whereas here they are not. The third type, which is very prominent in tissues, is a type where you don't see really a patterning by just looking to it. So what is typical here? Typical here is you have capillaries everywhere, but you also have highways everywhere. So if the capillary is something like the home, and then you go to this street, and very fast you are on a highway. So the way to this home and from this home is very fast. So you have a kind of a combination of this one and this one. You have capillaries everywhere, but you still have a very efficient supply and drainage system. But that has to be embedded in the same space. And as you see here, this I could draw on my computer the same here. I tried to do this, but I stopped after a while because everything looked ridiculous, and I took a real network instead. Because it's not so easy to create something like this. And obviously that's a very relevant mechanism, and I will give you a little bit of my ideas how these types of networks could be generated. Just to show something, how it looks in vivo, this is from a skeletal muscle, and this is on the capillary level, and you see that you kind of have capillaries everywhere, but if you look a little bit more from the distance, you see that you not only have the capillaries, but you also have, doesn't want to start again. You not only have capillaries, but you also have these hierarchical vessels to drain and feed these. So when you take this together and then go back to what we call angiogenesis, you might see that there are different types of angiogenesis and also angiogenesis research. The most frequently used paradigm of angiogenesis is a so-called sprouting angiogenesis. There's a huge literature on that, and I will come to that, where from existing vessels there are these kind of blind-ending sprouts, and they wander into the tissue, and VEGF is a very strong stimulus to create these vessels, and then they have to link to existing vessels to create some flow. In grown-up, this is very typical in wound healing. It's also typical in inflammation and tumor growth. It's driven by hypoxia and growth factors like, for instance, VEGF. The resulting networks have to be heterogeneous because a sprout doesn't know where to go. You have to consider that other than in this kind of very engineer-like network, there's no one having a blueprint and looking whether the result is anything similar to the blueprint. These vessels have to decide on their own, if you wish, where to go. So it's a very stochastic and randomized process, and therefore the speed is relatively low because you try and error all the way. And also you have to create many vessels which never make it to the final network because they are just at the wrong place, because there's no way of guiding them to the right place. But this is very, very heavily studied. The question is that really right? Do we focus properly in our science? It is very heavily studied because you can talk a lot about the angiogenic or anti-angiogenic factors. You can see it in a in vitro setting. You see the sprouting cells, and it's a very fascinating process. But the question is it the only process, and is it the most relevant process? It is a relevant process, but I think there are other processes which would deserve much more attention. One process which was established by the burn anatomist and Jonov and Burri, and is called intersusception, or easier splitting growth, is a process where you have an existing vessel, and due to some stimuli which are not well defined but probably flow plays a role, it's splitting into parallel vessels and creating a parallel expansion of a network. Burri said for the lung, for instance, it's completely impossible to have this explosive growth of a lung in embryology without a process which is not based completely on stochastics, but rather has a more logical expansion. And so it's probably a process which is, during growth, is very, very relevant for many tissues, like the lung, maybe the liver. It's certainly not the case for, let's say, the brain, because the brain first starts without vessels, and then you have sprouting going into it. So you have to define the tissues you are talking about. What are the advantages here? You have always blood flow. Here you have these blind ends where you have no blood flow, but here you have continuous blood flow. So the system has this kind of very neat property that it works while being constructed. If you look at the Autobahn or a house building, the bad thing is during construction of the Autobahn, you have a problem. During construction of the house, you can't live in it. That's the same like here. But here you can continuously use it, and it's growing with your needs, more or less. And therefore, it's a relatively fast process. It can be continuously remodeled based on functional feedbacks, whereas this one has no functional feedback because it doesn't serve any functional purpose. So this is a very interesting but grossly understudied mechanism. The reason why it is understudied, it's very difficult to follow it up in vitro. So the anatomists, they did corrosion casts and electron microscopy, and they show these pictures which they then put into a cartoon, and they say it came from here to here to here. But these were 3 animals. So it's very, very difficult to really follow it in real time and prove how it's done and study the mechanisms. On the other side here is a mechanism which is also very prominent now because it's studied intensively in zebrafish, and that is a patterning of the major vessels where you could talk of a genetic hardwiring. This may be the dorsal aorta and the intersomatic vessels, and they are patterned, and as you know, you learn these vessels in anatomy. If you are studying medicine, the vessels which you learn by heart, where you have to know the names, these are probably vessels which are created by a fixed blueprint, genetic type of blueprint. But the number of vessels which you don't learn by name because they are different in any 2 people, they are the majority of the vessels. So this is probably very relevant for the genetically hardwired big vessels. It takes a long time because it also goes without a functional feedback. When you are creating these intersomatic vessels, you don't have blood flow. So first you have to more or less guide them by some kind of growth factor gradients and repellents and attractant molecules into a space, but without having blood flow to tell you whether it's right or wrong. And then in between is something which is also very interesting and might be really relevant for the lung, but it's not yet in the literature. It's a term which I want to coin on that, which is emerging vasculogenesis or angiogenesis. And that's a system where you have a hierarchical type of tree for the arteries and veins, and you have a mesh to link them. So on one you are more or less combining effects of both types of angiogenesis. And I will show you one or two pictures for this. The good thing here is continuous functional remodeling. You have no non-perfused vessels, so the system can grow and work in parallel. If you consider, for instance, the chorion allantois membrane or the placenta of an embryo, the embryo is growing, this supplying tissue is growing, but there are no building periods. So the embryo cannot say from day 5 to 6, I stop it and I come up with a new thing. No, while growing, the system has to be completely functional all the time, but growing very fast. So you have two very difficult to combine restraints. It has to grow very fast. It has to be functional all the time. And that can be done by this. Here you see that from the chorion allantois membrane again. It's a picture by Valentin Jonov. Here you see this kind of capillary mesh, which is completely non-hierarchical and has a typical mesh size. And has a very high flow resistance, but good diffusion properties. But to feed and drain it, you need these larger vessels. And as you see here, they emerge more or less from this homogeneous isotropic background of small vessels. And then they create a hierarchical vessel. But if you look at this intravital microscopy here, these are all the hierarchical vessels. But if you would look into these spaces here, they would look like this. So this yellowish thing is a complete mesh of vessels. And this allows to grow in a kind of self-similar fractal fashion and very, very fast without any randomness, in the sense that you first put vessels and then you see, okay, this is not good, I take it out again. This is all workable and it's therefore very cost efficient. So now I come to the sprouting again, because obviously the sprouting stuff is the most researched. I was looking into publications in PubMed with a title term Angiogenesis. That was March 2014, so maybe there are a few thousand more now, and these were like 17,000. And here you see from Carmelite or Rizau the accepted general scheme. So you have some progenitors or whatever, and you create a primitive vascular network, and then by some miraculous means, this completely non-functional network is transferred to a what they call mature vascular network, or here they call it remodeling. And there comes the term pruning. But if you look, I mean, here it looks like, okay, this is one relevant step, this is one relevant step. But if you look into PubMed, how many people publish on these two relevant steps, you see that like 17,000 publish on the first step and 146 publish on the second step if you both look only in the title, and if you allow even the abstract, you come to 1,000. So in principle, we know a lot from here to here or from here to here, and very little is known from here to here. So we could ask, yeah, is that really relevant? If we have vessels, it's good. My comparison is always, okay, if you want streets, call a helicopter who drops concrete to the landscape, and then you have more or less what streets are made out of, and that's nice. But you know that without remodeling of the concrete, it will not really help you. And that is probably also the reason why the earlier pro-angiogenesis trials did not really work, because just having a lot of small vessels doesn't make the tissue very happy. And the reason for that is obviously Poiseuille's law again. Here I have networks. This is just the outline of the vessels, and this is the corresponding oxygen distribution. And for instance, if you compare these two networks, this is the artery, these are the veins, here the artery and the veins. If you look only at the right side, you would say, okay, these networks are more or less identical, yeah? Because you don't see a big difference in vessel diameter. But there are some vessel diameter differences, and you have to imagine in Poiseuille's law, it's a fourth power. So small changes in diameter make big changes in perfusion. And if you look to the functional outcome, here you have a good oxygen distribution, here the oxygen distribution is so-so, despite the fact that the diameter distribution is very similar. Here, another example, if I would give you only these pictures and say, are they different or similar, you would say similar, but this vessel is alive, this tissue is alive, this tissue is dead. So with only small changes in vessel diameter, it doesn't help you that you have a lot of vessels. They are just not functionally adequate. And you all know that from the tumor field. Tumors have different vascularity, but some tumors have very good overall number of vessels and even very good overall perfusion per 100 grams of tissue a minute. But some tumors have a very strong fraction of hypoxic local areas because if a perfusion is just not right, and then they look more like this. You have a lot of hypoxia, you have some areas where you are well oxygenated. So forgetting about the remodeling and pruning, I think, is a major drawback in the current scenery of angiogenesis research. If you look at stepwise, like in the Rizau and Carmelite papers, you have these two different settings. One setting is vascular genesis, where you create vessels from progenitor cells, hematopoietic progenitor cells in the tissue, so you don't have pre-existing vessels, they just created. In angiogenesis, you have pre-existing vessels, and then you have sprouting. But in both cases, you have this kind of unordered, non-functional test network. And then what you do is, from here to here, you reduce the number of vessels because you take out everything which was not working properly. So it's a very costly mechanism because you have to create like 3 times of what you will be ending it with, and you all know it from wounds. If you have a wound, if you have a scratch wound, it will be pretty red for quite some time, and when it's healing, it's getting lower to the normal degree of perfusion, or if you have a scar, it will even get white. So what is the problem here? The problem is stochastics. If you have a symmetric network, you need one dimension more than the dimension which you want to supply. That maybe sounds a little bit mathematic, but in principle, if you want to embed the vessels in the plane here, which you want to supply, you inevitably have short pathways and long pathways. Only if you allow to supply a plane, or here it's more or less a line, if you allow one dimension more, then, okay, you can supply each point in this plane with exactly the same feeding and draining network. But you all know that for a planar system, you could add one more dimension, the third dimension, but if you want to supply the heart or the lung, where do you add the fourth dimension? So it's a little bit difficult, and therefore you get this dimensional problem. The other problem is that you have the stochastic generation of new vessels, and I don't want to go into detail, but these two things create heterogeneity of perfusion and heterogeneity of oxygen. Then you could say, okay, we may install some feedback mechanisms to reduce that, like where you have too much oxygen, you restrict the vessel diameter, but you will never transfer a stochastic network into these engineer's networks with respect to efficiency. And maybe I skip this theoretical one in sake of time and go to the graphic distribution. So for instance, if you look at the network like this one and you plot any parameter, here we plotted flow velocity versus shear stress, or wall shear, sorry, this is flow velocity versus pressure, and this is wall shear stress versus pressure. And the red ones are the arterioles, the green ones are capillaries, and the blue ones are the veins. When you go with pressure through the network, so the blood flow goes from here to here, you see that the variability of both flow velocity and shear stress are very, very big. Many people investigating isolated vessels say the arterial shear stress in humans is 10 dynes per square centimeter, and the venous shear stress is 1 dyne per square centimeter. Here you have at least 2 orders of magnitude variation, so 1 to 100. It's by no way it is true to say the arterial shear stress is this and this. So you have a lot of heterogeneity which you would not have in any engineer type of network. And we used a computer model to analyze that, but to again show it more in a graphical term, here are networks and they are color-coded for different of these signals which vessels use to adapt their diameter. So for instance, this is a metabolic signal which could be oxygen or oxygen deficiency or anything derived from that. And this is a capillary bed, or is a part of the capillary bed. This is the arteriole, and obviously in the incoming arteriole you have more or less no metabolic need signal because the arteriole is very well oxygenated, and then you go down the way and the signal starts to build up. But if you consider the capillary bed, you have capillaries which have a very high oxygenation and therefore a very low signal, and you have capillaries which have a very low oxygenation and a very high metabolic signal. So the capillaries are not alike. There is no typical capillary. Then you could look, for instance, to hemodynamic signals, the shear stress. The vascular biology field knows that the shear stress is very relevant to determine vessel diameter. And you see that this vessel, which had a very low metabolic signal, has a very high shear stress signal, whereas this vessel, which had a very high metabolic signal, has a very low shear stress signal. So all these vessels have a kind of quasi-stable equilibrium, but with very different combinations of signals. So there is a lot of heterogeneity, but in the end, it converges into a stable overall situation. And that can, I think, help to understand what's happening during real angiogenesis, which should include remodeling and pruning. So if you start with a situation where you need new vessels because the diffusion distances are too long, the first step would be you create randomly sprouts. But these sprouts can be very unhelpful. For instance, this one here goes from a vein to a vein and will not have a lot of blood flow, whereas, for instance, this pathway here might be pretty helpful because it's connecting an arteriole on a vein. This one is a dead end, and so on and so forth. So this is not the end product. You have to first change vessel diameters, and then you will see, okay, when you consider this pathway, you have a lot of blood flow, so these vessels will get bigger. Other vessels which have no blood flow will get smaller, and then you have, and this one, which was not of much use, is also getting very small, and in the next step, you will eliminate these vessels. So you have these 3 steps: angiogenesis as a first step. Yes, it's a stochastic process, but immediately you have to start to remodel these vessels by changing vessel diameter, and then also take away those vessels which were not helpful for the end product. And together with my colleague in Arizona, we did put that into a computer model. Again, we started with a skeletonized vessel system like this one here, and then we allowed generation of vessel sprouts in areas which were hypoxic. So if you have not enough vessels like here, you have in the middle a hypoxic area. In the hypoxic area, you create a growth factor like VEGF, which is diffusing out from this area. If this factor reaches a vessel, it increases probability to generate sprouts. the sprout will not change the situation, and that's a problem. So you still have hypoxia because the sprout is not transporting any blood. So the sprout has to go on without any functional feedback. We call it a startup package. So it's like an economy. A company for the first 3 years is only burning money. And only after 3 years you can say, okay, if you still didn't reach the market, we take you out. But this sprout here did reach the market on the other side, and it did create a nice blood flow, so it may stay in. Yeah? So from here to here, you cannot control these vessels by the same mechanisms which you use afterwards. You cannot control startups by market rules. Only when they sell their products you can start doing that. And so this is the general scheme. And if you look what happens, then in the beginning you have a lot of hypoxia and a lot of VEGF. Then you create sprouts, you still have a lot of hypoxia and still a lot of VEGF, and therefore you are creating too many sprouts because the sprouts are not kind of helping with oxygen. Then in some areas you have a lot of oxygen. Here you see it's all yellow and orange, but still you have a lot of VEGF. Why that? You have very good perfusion here, but still angiogenesis signals. That's inevitable because VEGF has a pretty long diffusion distance, whereas oxygen has a very low diffusion distance. So in these areas, you're getting the VEGF from other areas, so you are still pushing angiogenesis. And you here have a overabundant density of vessels, and here you create new sprouts. And then the system starts settling down a little bit because you lower the VEGF level, and then you take out vessels which have, for instance, no oxygen delivery or no real blood flow, and you end up with something which is quasi-stable. It is fluctuating a little bit, so it's still reactive. And maybe that is, in the end, the advantage relative to an engineer's model. This model can always remodel according to changing needs, whereas if you have an engineer solution and you want more, then the engineer normally tells you, yes, build a new one, whereas this is adaptive. But if you look for the dynamics of that, you see that the classical scheme also from the Carmelite and Riesau paper doesn't really hold. You have angiogenesis in the beginning, but more or less in parallel with a slight phase shift. You have the structural adaptation and the pruning. It's obvious that you cannot adapt or remodel vessels which are not there. So angiogenesis has to come first and then structural adaptation and pruning, but they are grossly overlapping, so in principle they are a joint process. And you can use such a model to probe different deficiencies, obviously. So for instance, if you reduce the adaptation and pruning, you get a system which is workable to some degree, but it looks more like a foam rather than a real network. So you have these bubbles more or less, and it is not very efficient. On the other hand, it needs a lot of vessels. Or on the other hand, if you block different pathways, I did not talk about the conductive response, but here you see you get two vascular trees, but you get a avascular zone in the middle. So I think that combining the angiogenesis work of the typical type with looking a lot at the different molecules involved, together with the responses in adaptation, collateralization, and pruning, will hopefully result in much more efficient therapeutic approaches as compared to those which we have seen so far. So with that, I would like to thank you for your attention, and I'm open for questions.